Dear students, in this class we will be discussing about proteins. When you think of the food we eat, like egg, milk, fish, pulses, you are consuming proteins that help your body grow and repair. When you think of our muscles, our hair, our nails, all are made up of different types of proteins. Proteins occur in the protoplasm of all animals and plant cells. So, they constitute nearly 3/4 of the dry material of most living systems. So, chemically, they might be made up of amino acids joined together. But in real life, we can think of proteins as the workers and building material of our entire living system. Having said that, let us go into the chemistry of proteins. Proteins are essentially biologically macromolecules that play a central role in the structure and function of living system. They are high molecular weight polymers. In the last class we said they are polypeptides made up of alpha amino acids which are linked together by peptide bonds with a molecular weight of over 10,000. We have already discussed this in the last classes. Now, why is study of protein important? It is important because to form a foundation of more specialized branches such as biochemistry or pharmaceutical sciences, understanding of the chemistry of amino acid, peptide bond formation, denaturation, isoelectric point, classification and biological significance of proteins are all important. So first, let us begin with the classification of protein. So our classification is based on the composition of proteins. Based on the composition of proteins, proteins are divided into three. First one is the simple protein. Second one is the conjugated protein. And third one is the derived protein. So first one is simple proteins. Simple proteins are made up of only amino acids joined together by amide linkages. And these protein on hydrolysis gives only amino acids. There are many different examples of simple protein. First one is albumin. You already know about serum albumin. Then there is egg albumin, lactal albumin. All these are examples of albumin. Second protein is globulin, serum globulin or tissue globulin. Third one is prolamine. These are proteins found in the various plants, like for example zein from maize or gliadin from wheat. Fourth example is histone. Histone is a protein which is present in nucleic acid and hemoglobin. The last of the simple protein is scleroprotein, which for example keratin. Keratin is found in our hair, nail, skin. Then fibroin is found in silk. So these are some of the examples of simple protein. The second category based on composition is conjugated proteins. Conjugated proteins have a non-protein part in in their molecule. So in addition to amino acid part, they also have a non-protein part. And this non-protein part of conjugated proteins are known as prosthetic group or cofactor. Now, some examples of conjugated proteins are, first one, phosphoprotein. In phosphoprotein, as the name indicates, the prosthetic group present is phosphoric acid. Example of phosphoprotein is casein. Casein is the protein present in milk, or vitelline, which is the protein present in egg yolk. The second conjugated protein is nucleoprotein. And in nucleoprotein, the prosthetic group is nucleic acid, and it is present in the nuclei of plant and animal cell. Some examples are nucleosomes, which is a combination of histone and DNA, ribosome, ribosomal proteins and RNA, and viral capsids. The third conjugated protein is glycoprotein. Here, the non-protein part is a carbohydrate or its derivative. Example is the salivary mucin protein. The fourth conjugated protein is chromoprotein. These have colored, as the name indicates, they have colored prosthetic group, especially a pyrrole derivative. And example of chromoprotein is hemoglobin. So, in this prosthetic group, in addition to pyrrole derivative, there will be metals. For example, the metal present in hemoglobin is iron. So, then their cytochromes are also examples for chromoprotein. So, here we have studied 4 types of conjugated proteins: phosphoprotein, nucleoproteins, glycoproteins and chromoproteins. Then the last category of conjugated protein is derived proteins. Derived proteins are degradation products formed in various stages by the hydrolysis of simple or conjugated proteins. For example, if we hydrolyze proteins, first we may get proteoses, which can be broken down into peptones, which then converts into polypeptides, and that can be broken down into simple peptides. So, proteoses, peptones, polypeptides and single peptides formed by the hydrolysis of simple or conjugated protein is called derived proteins. Derived proteins. So, I hope you got a basic understanding of classification of proteins based on the composition. Proteins can also be classified based on the function and molecular shapes, and that classification is present in your test. Kindly go through the classification. We move on to the next very important topic in protein: color reactions of proteins. Color reactions of proteins are reactions which are used to identify proteins. So, here we will be learning about 5 most important color reactions. First one is the Biuret test. So, Biuret test is on adding dilute solution of copper sulfate. You take a copper sulfate solution, add this copper sulfate solution to an alkaline solution of protein, that is protein plus NaOH. If protein is present, then we will get a purple or violet color. This violet color is due to the formation of a compound known as biuret. Biuret is formed by the combination of 2 urea molecule, NH2, CO, NHCO, NH2. So please study the structure of biuret. It is important for you even in the analysis of organic compound part. So this is the biuret test. The second color reaction is xanthoprotic test. Here most of you might have observed formation of yellow stains on fingers while working with nitric acid, and that yellow color is due to the reaction of nitric acid with the protein present on our skin surface. When the nitric acid reacts with the proteins on the skin, it forms an orange colored acid called xanthoprotic acid. So that is why our skin turns yellow or slightly orange when we handle nitric acid. So in the xanthoprotic test, a protein solution is warmed with nitric acid. Then we will get a slightly yellow color. Now on making this reaction mixture alkaline, so when we add NaOH, this yellow color changes to orange color. So this test is known as xanthoprotic test. Third test is the nitroprusside test, and this test is given by proteins containing SH group, thiol group, especially cysteine. And when we react these proteins with nitroprusside, sodium nitroprusside solution, it gives a red color. Millen's test. Millen's test is used for identifying proteins with phenolic OH group. We react these proteins with Millen's reagent, which is mercurous and mercuric nitrate in nitric acid. Then we will get a precipitate which turns red on heating. So that is Millen's test. An example of a protein with phenol group is tyrosine. The next important test is ninhydrine test. When a pyridine solution of protein is reacted with a reagent called ninhydrine, a deep blue color or violet, pink or red color is obtained. This test is due to free NH and COH group present in the protein, and ninhydrine test is also given by amino acids. So we have gone through the color reaction. The last topic which we study here is the denaturation of protein. So we have all seen this reaction. When we put egg on a hot pan, it will turn white. The yolk will turn white. That is an example for denaturation of protein. So when heated or when treated with the strong acids, bases or reagents like heavy metal salts like mercury chloride or copper sulfate or picric acid, proteins are coagulated and they cannot be reversed back into the original substance. Hence, it is rendered insoluble and they lose their biological activity. These modified proteins are called denatured protein, and the conversion of a folded protein into a straight protein is known as denaturation. Denaturation, that phenomenon. Denaturation. Example of denaturation, we have already discussed coagulation of egg white by heat. So far in this class, we have learned about proteins, classification of proteins based on their composition, the color reactions of protein, and denaturation of proteins. In the next class, we will study the structure of protein. So till then, thank you.